Hello, I'm Dr. Kaveh and welcome to EaseGenetics. In this video, let's dive into one of the most important concepts in genetics, dominance. From the classic Mendelian rules to more complex interactions, dominance helps explain why we inherit traits the way we do. Knowing and understanding the types of dominance is very important, and it enables us to have a better understanding of the interactions between the alleles of a trait to identify individual genotypes and the inheritance mechanism of traits. When we think about dominance, we often picture Mendel's pea plants and his experiments with tall and short plants or yellow and green seeds and pods. Dominance is all about how alleles interact to produce a trait. But there are also other types of dominance beyond the dominant recessive model introduced by Mendel. In this video, I'll explain them all. Before we start, there is one important thing that I want to tell you. Remember, in all types of dominance, the phenotype of the two homozygous genotypes is two different things. So the phenotypes of homozygote big A differ from small a. They are two different things. Actually, in various types of dominance, what matters is that what is the phenotype of the heterozygous genotype. So various types of dominance determine the phenotype of the heterozygous genotype. I also want to clear three important facts. First, in every living organism, a trait follows a dominant inheritance pattern which is universal. This means that when Mendel found out that tall plants in green peas are dominant to short plants, this is the trait inheritance pattern, and regardless of where you're living on planet Earth, this pattern rules in green peas. Second, it can never be said in general that one state of a trait is better than another. For example, you can never say tall plants are better than short plants. Tall plants can be better in, for example, corn, but in crops, especially wheat, short plants are better than tall. And third, a trait's more commonly observed state is never necessarily an indication of its dominance. For example, in most countries, people like peas to be green. This doesn't mean that green color is dominant because we know it's recessive and yellow seeds in pea are dominant. Now let's start with complete dominance, the classic Mendelian scenario. Here, one allele completely masks or covers the effect of the other allele. For example, in pea plants, having just one big T allele is enough to make the plant tall. The recessive allele, small t, is only expressed when it's in a pair. This occurs when the dominant allele produces a phenotype that is identical in both heterozygous and homozygous dominant individuals. So as you see here, in complete dominance, plants with either big T or big T small t become tall, and plants with genotype small t will be short. Next, we have incomplete dominance, in which neither allele is completely dominant, and the result is an intermediate blend of the two different homozygous phenotypes. The classic example for this is snapdragon flowers. When you cross red and white flowers, you get pink offspring, and it's exactly a middle pink, just like mixing equal amounts of red and white colors, which gives you an exact middle pink, not light pink or dark pink. This is important because the following type of dominance is like this. Now, partial dominance is often used by mistake with incomplete dominance, but this pattern of inheritance refers to where the heterozygous phenotype is intermediate between the two homozygous phenotypes, but not exactly in the middle, but leans closer to one of them. This occurs when one allele is not completely dominant over the other, but still has a slightly stronger influence on the phenotype. For example, in some breeds of chickens, the allele for black feathers big B and the allele for white feathers big W show partial dominance. A heterozygous bird BW might have gray feathers but with a slight tendency towards either a darker or lighter shade depending on the degree of dominance. Now to codominance, where both alleles are fully expressed. When we talk about human ABO blood types, if you inherit an A allele from one parent and a B allele from the other, you'll have type AB blood. Both alleles contribute equally to the phenotype. Well, this is a classic example. Another example is coat color in certain animals, such as spotted cows, where both colors appear distinctly and simultaneously in the heterozygous phenotype. Have you heard of overdominance, also called heterozygous advantage? This one is my favorite. This phenomenon often results in enhanced fitness, survival, or performance of the heterozygous individuals in specific environments. A well-known example of overdominance is sickle cell trait in humans. Individuals heterozygous for the sickle cell allele HbA-HbS are more resistant to malaria compared to those who are homozygous for the normal allele HbA-HbA or homozygous for the sickle cell allele HbS-HbS. This provides a selective advantage to heterozygous in regions where malaria is prevalent. Another example can be seen in many traits in corn, especially plant height, where the heterozygous phenotypes are taller than either the two homozygous phenotypes. So which type of dominance is your favorite? Mention it in the comments. Let's see what is your taste. Now, let's take an overview of the 5 types of dominance I mentioned. If we place each genotype on a bar indicating the phenotype, as you see in all types of dominance, the two homozygous genotypes stand in 2 different positions. What differs in types of dominance is the standing or appearance of the heterozygous phenotype. Starting with complete dominance, you see the heterozygous position or standing is where the homozygous dominant is. In incomplete dominance, the heterozygote is exactly in the middle. In partial dominance, it's somewhere between the two homozygous, except exactly in the middle and somewhere leaning toward one of them. In codominance, the heterozygous phenotype stands on both homozygotes with a mixture of them, but separately and not blending. And finally, in overdominance, you see the heterozygous phenotype is out of the range of the two homozygous phenotypes, either higher than big A or lower than small a. Sometimes genes interact in ways that alter dominance relationships. This is called epistasis. So epistasis is a genetic interaction between 2 or more genes, while the interaction between the alleles for each gene also remains. There are various types of genes interactions or epistasis, which I will discuss in a special video, which I will call it genes interactions. So for now, I'll leave this for that. To recap, we've explored complete dominance, incomplete dominance, partial dominance, codominance, and overdominance. Understanding these patterns not only helps us decode genetics, but also provides insights into evolution, medicine, and agriculture. If you found this video helpful, don't forget to give it a like. Share it with your friends and subscribe to EaseGenetics for more engaging content. Have questions or topics you'd like me to cover? Drop them in the comments below. Thanks for watching, and I'll see you next time as I make genetics as easy as possible.